All right. Thank you all for joining once again. We are just going to recap from last week. So for last week, we looked at Genius Prime, how to install Genius Prime. And remember, it's 30 days free trial with a new email. So we'll have enough time to go through using Genius Prime for your bioinformatics solutions before it expires, before the 30-day trial is up. And also, we did look at Terra.bio bioinformatics resource page, the public health resources. And this is really, really important to familiarize yourselves with the resource page, because almost all pathogens that you are going to be working with, you can find the resources on the resource page to be able to do your data analysis using Terra.bio. Once again, for those of you who do not have a Terra.bio account, still working on the video that will allow you to set up your Google bucket account and be able to use Terra.bio with that free $300 credit for now. So let's start this week's course. So before we actually get into using these resources, bioinformatics comprises of two things: biology and informatics. You cannot do bioinformatics if you don't understand the biology, and you are going to be inefficient in actually doing bioinformatics if you don't really understand the processes. So it's good to understand the biology behind the informatics. And so let's go over that very briefly. And once again, the goal is because I do not want to show you how to do things. That is not the goal. A lot of videos out there, a lot of trainers out there will show you how to use the tools, will show you how to analyze samples, will show you how these things work, will show you everything you need to know about bioinformatics and data analysis. My goal is not to show you. My goal is to teach you, which are very, very different things. Teaching you means that I'm actually breaking down the reason why you are doing what you are doing and how that can be affected when there is a change. That makes you more independent so that you can actually do these things and troubleshoot errors when they occur by yourselves, because you will understand the fundamental basis for all of these tools and the reason why we are doing what we are doing and using these tools. Showing you will only make you memorize it. So at the end, you think you know it, but actually you haven't really understand how they work. So in the long term, you end up forgetting if you don't use them, because you actually never got the chance to learn it. You just memorized it. So I'm not going to show you. I'm going to teach you. So let's begin. Let me pull up the notes here. Let's see. So first, let's go over briefly what we look for when we talk about bioinformatics. Basically, what is it? Just to bring your mind and wrap your mind around the concept of bioinformatics and what we actually look at. Let's find the board, stop, breakout rooms, whiteboard. Okay. Then we change the name to Let's call it bioinformatics, biology workflow. So I just want to walk you through a workflow, or we are going to work through a workflow together about basically what we are looking at when we talk about bioinformatics on a high level. So all of you bioinformaticians out there, do not jump on me. This is high level. We'll get into the really nitty-gritty in depth when we actually start using these resources. So basically what happens? Let's take a scenario. There is a potential outbreak, or someone is sick. There's a disease, something has happened. So we can say someone is sick or ill. Let's make this bigger. Bold. Bigger. 16. 4. Let's make it even bigger. 36. All right. Someone is sick. What usually happens in a lab setting, two things. One, we may be able to have an idea of the sickness based on associations and based on what we call conceptual data. So basically, if it's an outbreak, is this outbreak happening somewhere else in close proximity? If someone, example of SARS-CoV-2 or COVID, if someone is showing symptoms of COVID, can we say that, oh, this person met with this other person who was diagnosed of having COVID, so we know it's COVID, or we suspect it's COVID. So usually we have an idea, or we would take a collection of the sample, and then we'll bring the sample to the lab to test. So we can call this, let's call this, we collect samples. And once we collect the samples come into the lab. We can say the lab processes the sample. So we collect samples, lab process the samples. The lab identifies by other methods. So there's a PCR method that will let you know what pathogen it is. So the lab identifies this, and the lab says, this is, what does this do? No, we don't want this. Let's undo the frame. Let's undo both of them. Let's go here. Recording stop. Recording begin. All right, so we are here and we said this is, so we have a situation where someone is sick. We collect samples and we bring the samples to the lab. The lab processes the samples and they do a first step diagnosis, usually by PCR. And we can say that this came back as positive for SARS-CoV-2. So this is positive SARS-CoV-2. So sample comes back positive for SARS-CoV-2. So we know the person is COVID infected, has COVID. And we are going to now go ahead and sequence the virus. And we are going to look at the genome once we do sequencing and basically look at the nucleotide. So what we do from here is, let's put another one here that says sequencing. So the lab will do sequencing of the samples. And then at the end of sequencing, the lab is going to see the nucleotide, the genomes. And this are shown in terms of nucleotide bases. So example, for this case, we'll have AAA, TTT, and let's do CCC. So this is our final result. Our final results are AAA, TTT, CCC. If I can put that even down. Let's do this. Let's change this to a different color. If you can. Yeah. So we have AAA, TTT, CCC. That was the result from our sequencing run. So what are we going to do? Obviously, the file we get from the sequencer isn't this clear, but we assume everything is moved on, everything worked, other processes have been completed, and then we have this sequence as our final. And usually it will be in a FASTA file, FASTA file. So what we look at is that we take this AAA, TTT, CCC sequence that we just did, stop recording, recording begin. So now basically we have our AAA, TTT, CCC as our sequence, final sequence from the recording stop, recording start. So finally, we have our sequences. And this is from the pathogen that we just sequenced, the SARS-CoV-2. So we have AAA, TTT, CCC. Once again, high level, what we do is, since we know that this sample is SARS-CoV-2, we are going to go on to the next step and compare the genomic sequences. So basically, the genetic sequence of this SARS-CoV-2, which is AAA, TTT, CCC, with the known wild type sequence of SARS-CoV-2, or with a known sequence of SARS-CoV-2. So let's say, example, if the known SARS-CoV-2 sample, let's do this. This is known SC2 SARS-CoV-2, and we usually call them a reference. But for this basic introduction, we just call it sequence to keep it consistent. So we'll put this known SARS-CoV-2 sequence here. And then we know that the known SARS-CoV-2 sequence out there, let's say, is AAA, TTT, CCC. So what we do is, up a little bit. We compare now our sequences to the known SARS-CoV-2 sequences out there, also called a reference. So in this example, the sequence that we just sequenced from our sick patient is AAA, TTT, CCC. When we compared this to the known SARS-CoV-2 sequence out there, which is also AAA, TTT, CCC, now, because the sequences are the same, 100% identical, we can say that our sequence is or has the same similarity to the non-SARS-CoV-2 sample. So if the non-SARS-CoV-2 sample that we use for our reference was, let's say, B1, then we know that our sample is B1. If the non-SARS-CoV-2 sequence, let's say if this one is, let's say if this was known to be Delta, then we can say that our sample is also Delta because it matched. So that is basically what we are doing when we are calling these names. We are just comparing our sequences that we just sequenced to a known sequence for one aspect of bioinformatics. And that is usually called a reference-based assembly. So reference meaning that we already have a reference. We already know what we are dealing with. So in this case, they're the same when we compare it to a known reference. There are some other times where our sequence doesn't match the known reference. So let's maybe change the TTT to a GGT. It's bigger. So now we see that the known reference sequence, which was AAA, TTT, CCC, known as Delta in this example, our sequence has a different, or we'll call it a mutation, where now we have AAA, GGT instead of TTT. So two of the T's from the original one has now been changed to a GG. And I'm just going to circle that right here. Now that has caused a change or now we are seeing a difference between our sequence from this patient that has that was positive for SARS-CoV-2 to this database or reference known sequence. So we can take a look at the literature and see what this GG what effect it may have on the lineage of the SARS-CoV-2. So this we looked it up and we realized that when TT is replaced with GG, now it is no more Delta. It is now you can call it Omicron. Let me pull this up. And here we go. So basically, whenever we do a reference-based, and I'll put the name here on top, we call this reference-based assembly. For now, let's use this word. So this is what we call a reference-based assembly, where we know what pathogen we are dealing with, and then we are able to compare it to our sequence. And then if it's the same, then basically we know it is the same as our sample. So example, in this case, if it wasn't SARS-CoV-2, and let's say it was influenza, if our sequence was matching 100% to the reference influenza, our sample is influenza A, matches 100% to the reference influenza A, then we know our sample is influenza A. But then if we realize that our sample doesn't 100% match to the reference of sequence of influenza A, then we know our sample is not influenza A. And then we try to figure out, based on where the changes are in the nucleotides, what those could be. And we can all match it to, let's say, another reference like influenza B or C or any of those and see if it matches 100% to it. So basically, that is how we do assembly-based. And this can be the same for dengue, any pathogen, any virus. HIV-1, if sample matches, it's HIV-1. If it doesn't, we find out what it is. Most of these pathogens have references out there. So nowadays, it's very easier or less complicated to identify what pathogen you are working with. And once you can confirm it by PCR or by any other methods in the lab, and you know what pathogen you are dealing with, you can go ahead, do the sequencing, get your sequence file, and then compare it to the reference to see if it's the same. And if it's not the same, are there any differences? If there are differences, what are the differences occurring and what do they mean? So that is in a nutshell what reference-based assembly for bioinformatics is. And once again, going from once you get your sequence to actually getting the real sequences, where you can do these comparisons, there's a lot more process involved because once you put in a sequencer, the results that come out from the sequencer are usually FASTQ files, FASTQ files. And you have to process the FASTQ files in order to get a FASTA file where you can use to do this alignments. That's what we call them. We are just aligning them to see if it's similar. Now, second scenario, what if there's an outbreak, but you don't really know the source? Or in this case, you don't really know what pathogen it's causing the disease or the outbreak. And there's also a different bioinformatic assembly. So we have the reference base where we know a reference and then we use it for a reference. And then we have the other assembly where we have no reference because we do not know what pathogen it could be, or there could be multiple pathogens in there. So doing a reference base isn't going to really help you. And you can do this usually with mixed samples, metagenomics, mostly for like bacteria, because there's a lot of bacteria usually present in a species in one sample, so multi-species sequencing or assembly. And the word that we use for that is de novo, D-Novo. So let me create another section for when we have to do bioinformatics on things we have no idea about. So this is de novo assembly. First one is reference-based. Second one is de novo assembly. De novo meaning we have no idea. So someone is sick, just like the first one we did. So we can copy this one and copy that one too. That one too, and then just copy, and we can come here and paste it here. Because the scenario is still the same, except now we have no idea what is causing the disease. This here. Yep, so de novo assembly, someone is sick. The lab takes samples. The lab processes the samples. This case, because we have no idea, we can either do a PCR to figure out what might be in there, but that's going to be very difficult. So, or we may want to see what the profile, what everything that could be in this sample or could have caused this. And this is usually when there's let's say a dead animal that we want to figure out what caused the death, acute febrile illness, AFI for short. So that is like an illness that can be caused by multiple pathogens. So in that case, you don't have one, you have multiple. So you may want to do this de novo assembly where you basically take your sample and you're going to go through that. So the sample is processed, we sequence, but in this case, we sequence everything in the sample. So just do a line that goes here, and then we can say we sequence everything. We sequence everything, and now we have everything in our sample. We could have all kinds of bacteria, we could have some viruses, we could have some fungi, and we have everything. So we take the sequences. So just like the one we did before, so this can be AAA, TTT, CCC, GGG. And instead of comparing these sequences to a reference, because we have no idea, And this could be a lot of pathogens. So there isn't one pathogen where we can say, let's take one reference pathogen to align to our sequence. We align all of our sequences from our run to what we call databases. So databases contain a lot of, you can call them references, a lot of references from different pathogens that are already in the system. And then our sequence just goes in there into this database and does a full database alignment to everything in that database to see if it will align to some of the pathogens in there. So this is usually done by a database, and call this database. The database, let's use the word alignment. Alignment. And this would be multi-pathogenic. Let's use that word. So it will be multi-pathogenic. So first, we don't know what the pathogen is, or we suspect multiple pathogens, or we want to detect all the pathogens that could be in there. So usually bacteria are usually mostly what we usually do de novo for. Some viral pathogens that are either multi-pathogen or co-infections, we can do this same thing for. But mostly for bacteria or other non-viral pathogens. And so what we are doing is that someone is sick, or we find something, or we want to detect what is in a sample. The lab processes the sample. Once again, there are a lot of steps. I'm just skipping all the steps to giving you the main points and ideas. Once we have the sample processed in the lab will go ahead and perform sequencing. Once again, they sequence everything because we are not, we don't have any specific pathogen in mind. We want to see everything. So they sequence everything. And then once they sequence everything, now they have the sequences of everything. Because you have a sequences of everything, now, instead of comparing your sequences, just like we did before, to a reference, which is only one known pathogen, we want to compare it to a multiple known pathogens. And this is usually set in a database. And one of the most common database that you've probably heard of is NCBI. I'll put this in here. NCBI. It's backwards. It's NCBI. So NCBI is a database that has a collection of a lot of pathogens. So viruses, bacteria, fungi, all of them. And there are others out there that you can explore at your own leisure. So, but it's an example. You take your sequence and then you align it doing with this de novo assembly. Once you actually assemble it, then you do the alignment to usually when you're doing NCBI, we'll say blasting to all the sequences in the database. And your sample will come back picking up usually by proportion. So at the end, you will know that your sample has 50% viruses, 20% bacteria, maybe 10% fungal. And then you can actually go into more details to know or to see what type of viruses is in your sample, or what the percentage of virus is. So you can say, can have maybe some, or even bacteria. So it gives you and like a broad spectrum of what could potentially be in your sample when you do de novo assembly. So yeah, so first let's recap. What you're going to be using or doing as we go through this course are going to be two things. The first one is going to be a reference-based assembly. And what that means is that we actually have an idea of what pathogen we are working with. So in this case, you know it's SARS-CoV-2. So once you go through your data and you have your sequences, you will align it, or you do your reference-based assembly, and you would assemble it using a reference for SARS-CoV-2. And then you can, based on that, you will know if it's changes in if it's the same, 100% similar to the reference, or if there are any differences and what those differences mean. So take a sample, but we have an idea of what pathogen is in there, and want to see the variation of that of our sample, if it's the same or not the same as the pathogen. Reference-based. And then there is also the other one that we'll tackle in this course called the de novo assembly, which is also known as multi-reference. So basically, you don't have one reference. That's because your pathogen or your sample contains or has the potential of containing multiple pathogens. So as a result, we do an assembly called de novo assembly, where we once we have our sequences, we just blast it. When we're using NCBI, we say blast. But I'll use the word align, since that is what we use for the first one. That way I can keep the train of thoughts together. So basically you take your sample and then you align it to all of these other pathogens in this database called NCBI. And the goal is to see how many of your sequences actually align to the different pathogens. And at the end, it will give you a summary. So you get a summary of how many of your samples are viruses, how many are bacteria. Those that are viruses, what kind of viruses did your sequence match to? Did it match to some flu? Did it match to some SARS-CoV-2? Did it match to some HIV? A lot of them, RSV. So, and if it's bacteria, did it match to, is there some Klebsiella in there? Is there some Enterobacterus in there? Are there Salmonella in there? It tells you what your sample could have matched to. And then it's up to you, the bioinformatician, to use other resources and other tools to zero in more and to focus more on what it could be, because now you have a ton of things that could potentially be, but there are ways to rule them down to a few possible ones. And we'll go through all of that in this course. So here we go. Once again, thank you for following through. Now you have some understanding of some of the concept with the bio and then the informatics aspect to how we are going to tackle and answer these pathogen data analysis or concerns that we are going to be working through in this course.
Don't forget to hit the like and subscribe button below so you can get notifications when new videos are uploaded.